Research Forester with the U.S. Forest Service Northern Research Station.
I'm Mark Abney with the Natural Resources Conservation Service. I'm the Assistant State Soil Scientist here in Missouri. And John and I, and Jorge's here too, we're out in Basket Forest, and you can see that in the background. We've been roasting some mark by the fire and having a great time waiting on you all to come to the virtual tour. And today is a really awesome day to tour Missouri, and you're going to see some really unique things that were developed here in Missouri. When you go to the Dooley-Miller plots, it's where the universal soil loss equation started, and we're still using that today. When you go to Sanborn Fields, where they discovered antibiotics, and we're still using antibiotics today. And when you come to Basket, you're going to see a research area that has 76 plots in it. And of the 76 plots, we've sampled well over 50 of full depth, full soil characterizations with biology and surface replications of 3 on each site. And it was a tremendous effort by a whole bunch of people. And we are glad that you are here. If you want more information on Basket, you could email John or I, either one. We've got biological profiles. We've got all the lab data for all the pedons. And there's still more research work going on. So then enjoy the power washing demonstration. I like power washing. It shows us a lot, and I think you'll enjoy that video. So John, you got anything else to say?
Yeah, so the way this field trip will work, as we said yesterday, is we'll first play our introduction video for you so that you can get a little overview of what the field trip's about. And then it's on your own to watch all of the videos, the Basket tour, the tours of the Soil Health Lab. There are 2 videos that give you a little history of Sanborn Field and its significance and what's going on there today, as well as the video on Dooley-Miller. So you watch that on your own. Mark and I are going to check back in with you at about 1 o'clock, and we'll have a session where you can just write down all your questions that you have, and Mark will be happy to answer them for you. And but starting at 1 o'clock, we have an hour and a half put aside to answer any questions you have. Doug Wallace, who is also a part of our field trip, you'll see Doug in the videos this morning. Doug will also be on Zoom to answer questions about some of the vegetation and the ecological site data that we've generated, and also can link back up to the work that we're doing at Basket.
I'd like to just, as a final thing, say this has been a virtually awesome experience, and we are virtually worn out. And we want you to know that tonight we will be celebrating at the virtual banquet that you won't be at. So we'll be enjoying that. Not really, but anyway, have a great time today as you tour parts of the Boone County. And we are glad that you are here.
Yeah, thank you so much. We'll see you soon.
I think we're live. So probably like a lot of you, I've been going to these regional conferences for years, and the thing I remember looking back are the field trips. I mean, it's just great to have an opportunity to actually walk on the land and see the soil, see the vegetation, and to have those conversations about digital mapping versus traditional mapping and classification and on and on and on, and getting to know people. So in our case, this year, 2020, bigger forces prevented us from doing a field trip, an in-person field trip. But nevertheless, Mark Abney and John Cabrick and PQ and many others have still, I think, put together a good story. And so that's what Mark is going to do now, is to give an introduction to what we'll see on the virtual field trip. So Mark, please take it away.
Sure. It's right.
Okay, can everybody see this?
We can see it, but if you can put it in presentation mode.
That work?
Yes.
Is that good?
That is good.
Okay. Okay, so I guess welcome back from break, and we're going to talk a little bit about the tour tomorrow. And there's a lot of things in the tour other than the basket thing, and all that will be on demand. You'll be able to go and see all the basket pits and look at the ecological sites. You'll also be able to click on a power washing demonstration, and let me tell you right up front, it's a wash. There's other things that you can look at on that tour. So them are on demand, so you just go to them as you want. And at 1 o'clock tomorrow, we will take questions and answers. So we'll be in the room. John and I will be in the room together. Doug Walls will be on remotely, but we'll all be here to answer questions that you might have about any of the things that you're going to see on demand tomorrow. Later this afternoon, you'll get an email that will have an agenda for tomorrow that will take you to the links to all the tours. So there's Sanborn Field, Dooley Miller, the power washing, this video we're seeing here will be shown again before the tour, the basket tour. So all of them are on demand. So let's get into the basket tour itself. This is a proximity map, so you kind of know where we're at. Columbia is to the north. Jeff City, the capital of Missouri, is to the south. That river over there to the west is the Missouri River. That's the La Source. And so we are kind of in a breaks area. We'll look at the next map, which is the geology of the area. You can see we got a lot of geologies coming together here. Right there in the middle of the state in Cole County is where the capital is, in Jeff City, Missouri. Just to the north is Boone County. That's the county that Columbia is in. And in between, right in between Boone and Cole, if you would just put a little dot halfway in between, that's where Ashland is. And we're just a little bit east of Ashland, probably 6 or 8 miles east of Ashland. And you can see we'd be in that blue Mississippian zone, but also on the north side of the basket, we've got a little bit of glacial till coming in there. So the glaciers come down probably even further than in the area of the Missouri River, but a lot of that was till was eroded away. Yeah, I don't know if you can see that. Yeah, okay, Cole, Boone, and Ashland. Oh, you have to have it down there. Okay. So Cole, Boone, and then Ashland would be about right in there where we're at. So that's that Mississippian geology that we was talking about. Here's a block diagram, and this block diagram is not as elaborate and fancy as the Dillon Bodette stuff, and it's not animated or anything. I'm old school. This is the ones like we used to make and put in the old manuals. So I modified it a little bit to talk a little bit about the stratigraphy there. And these are the sites. Site 1 is on a ridge top just off of this whole and just across the road to the east is the agriculture. This is site 1, and A would actually be, the road is about right here on this ridge. Just across the road to the east is the agricultural site.
Mark, I'm sorry, I'm going to jump in here for a second.
Is an ag site. I think it's also a reference to site 1B is the same site, and then this is site 1A. So we used one soil pit for both them sites because it's actually the same soil. Site 2 is on the shoulder. Okay.
Oh, we cannot hear you.
Oh.
What audio is going off?
What?
Your audio is going off.
Mark, I'm going to jump in here for a second, but your audio is going out. And so we missed part of what you were presenting. If you want to go back maybe a minute ago and talk about the slide that you're on right now.
Okay. So we had the audio up until I started this slide.
Approximately.
Okay. So this slide to start over is a block diagram, and it's old school. It's not elaborate like the Dillon Bodette animated stuff. So it's what we used to put in our old soil survey manuals. I've written on it and kind of arrowed to some different things I want to show. So this site 1 is on a ridge top, and this whole transect that we're going to be looking at is moving to the west down slope towards the bottom. Site 1 is at the top of the ridge, and it's on a Weller soil, Weller-like soil. Site 1A is across the road. The road runs right along the ridge top. And just across the road is Site 1B. And to the east, and Site 1B is in an ag field. It's the same soil, so we just used the Site 1A pit. Site 1B is also referenced sometimes as Site 5. So I don't have that on my map, but it's just across the road, and it's an ag site. And you'll see in the ecological site descriptions that John and Doug do how this site differs from Site 1, which is a reference state site. So then we go down slope to the west, and we get on the shoulder, the upper shoulder of the back slope. And Site 2 is on a barley-like soil. And then moving on down, come out on what I call a bench. The limestone is holding this area up, and it's benched out, and there's some loess that's still left on top of the bench. And so normally we'd find Clinkenbeard, Roachport, Bonfim soils in this location, but Site 3 has quite a bit more loess on it than that. So it's kind of a hybrid between a Weller and some residual type material. But, it's a nice look. And then we drop down to Site 4, which Site 4 is probably more like a Dameron, but typically they map Jemerson and Cedar Gap in these positions. And so then to look over at the stratigraphy over here, on top we have Peoria and Luss. And then there's a lithologic discontinuity that shows up in the Luss soils, and below that is the Roxana Luss. There's a lot of controversy on these Luss deposits. We're pretty sure the upper ones, Peorian, the Roxana, is where the controversy is. So it could be Loveland. We think it's Roxana, but then just below that we have residual soils. And also on these slopes, we have colluvial material. So the residual soils are red soils, red clay soils. So we'll see some of them. And then some of them have colluvial materials on top of them. And all these residual soils formed in the Mississippi and limestone and dolostone. So that's pretty much a stratigraphy of the area. In the bottom, of course, we have alluvium, a lot of it's skeletal. And so we'll move on to the next slide. And then I'm going to let John start talking about this slide, but these are the plots in the basket area.
So hello, this is John Cabrick with the U.S. Forest Service Northern Research Station. I'm going to tell you a little bit about some of these plots. You may be wondering why we're working at the Basket Research Center, and it's because we have a whole variety of research plots that have been examined. Some of them go back to the 1960s. The plots shown on this map in green, like you see here and here, and scattered throughout this entire area, even up in the northeast corner of the Basket area, are part of a network of plots that were established by a biologist at the university back in 1968. His name was Rochow. And he put these plots in really just to study vegetation. And so since 1968, about every 12 to 15 years or so, a team of scientists might come out and re-inventory these plots, and we've been able to track how the forest has changed over all these years. Now, one of the things that's been absent from these plot work is the soils information. Occasionally, someone will take some surface samples, have some analysis done, maybe measure the pH or exchangeable bases. But other than that, there's been very little soil information that's been collected. And we saw this collectively as an opportunity to examine the soils and use this information to look at some of these long-term successional changes that have taken place in these forests, but relate it back to soils and site information in a way that hasn't been done before. Similarly, the plots that are shown in yellow, red, we'll call it sort of a purple, and also in blue are part of another study that's been going on at the Basket area. And in this case, this study examined the effect of past land use on soils and also on vegetation. And so in this study, the plots that are shown in yellow and in red are areas that used to be farmed up until the mid-1930s. And about that time, the federal government purchased some of that land. There were 17 different farms. You'll hear about this tomorrow on the field trip. And they were consolidated into federal purchase units. And in 1938, it was leased to the university, and finally, at some point in the future, it was officially transferred to the university. But it's been under the university's ownership or watch since about 1938 to do some research in this area. And so, in these plots shown in the yellow and in red, they were the old fields that were growing back into native vegetation. They're full of old field invaders, cedar trees, elms. You'll get a little view of that tomorrow. And the plots that are shown in the purple and blue are areas on the same soils,
but that have always been in native forest. And so that's given an ability to examine how that past land use, that past farming, and then abandonment, and then reforestation has affected the properties there, the habitat there, the wildlife usage and so forth, a number of different areas that were being studied. But we have those reference states also to that forest condition. The way the study was set up, they were also paired by soil map unit. So all those in yellow were agricultural barley. They have that ag legacy. All those in purple were also barley, but they have the forest history. The reds were all weller soils in agriculture. The blue plots are all wellers that have always been in forest condition. So you'll get a little sense of that tomorrow. This picture here is a schematic to give you a sense of what these plots might look like on the ground. This is a picture of the basket area. The red circular area here is supposed to be a plot boundary for measuring vegetation. The original plots in Rochelle and also in the new study use the same design, so it provides comparable data. This is a 5th acre plot for the overstory. It has a radius of about 52.7 feet approximately. And within that, they collect data on all the trees greater than about 4 or 5 inches DVH, diameter at breast height. We also have smaller subplots that are nested within those for measuring vegetation. And around the plot center, they actually have little pieces of wire with tags on them that they place near seedlings that they encounter during an inventory, and that gives them, the scientists, the ability to go back and track the fate of those individual trees over time, so that we have a really good picture of what's happening with all the different components of the forest. And then for our work with the soils, we didn't want to disturb the vegetation in the plot since it was measured carefully. So we would come through with an excavator. We were digging soil pits adjacent to these 5th acre overstory plots. And what we judged to be the same soil, we get to do a little inspection of the soil with hand tools, or we look on the same to make sure that we were on the same elevation. And we thought we had the same soil that occurred in the plot, and we would do that. Plus, we also took small samples of the surface with a 5 centimeter diameter ring that to a depth of 5 centimeters. And those rings are shown here with these little yellow circles nested within these plots. We took one on the upslope, 30 feet north of the, or 30 feet upslope of the plot center. We took one at plot center, which is shown here with this blue sun. And then we took one 30 feet below plot center, so that we had a couple of samples within each plot. And those were also sent to the lab. All of the samples that were sent to the lab received normal characterization, so particle size, base cations, pH, base saturation, as well as cation exchange capacity, the normal things that we do for characterization. But we also had the samples analyzed for the biological measures, so the phospholipid fatty acid analysis, which not only looks at total PLFAs, but also those that are attributed to fungi, bacteria, gram-positive bacteria, gram-negative bacteria, the full battery of biological measures that the Soil Health and Assessment Center here at the University of Missouri is capable of analyzing for. And I think, as Mark talked about in his talk earlier today, this is the new thing that we're doing in soil survey that's giving us a lot of insight into these soils in ways that we haven't routinely done in the past. So we want to show you what some of these soils might look like. So in this picture here, we're standing on a shoulder position. And if you look on the right side of your screen, you'll see what a typical soil might look like on a shoulder position. It has a little bit of a remnant lust cap that quickly starts to grade into these hill slope sediments. You can tell when you're in the hill slope sediments because you'll begin to see these coarse fragments Most of it is chert that was weathered from the underlying limestone, and it appears as gravel size or cobble size particles. And with depth, when you start getting into that residuum below, you'll start to see chunks of bedrock that are actually in that limestone residuum in that profile. We've had a large crew of people come in and work with us on this various sampling. This shows you a picture from a ridge top, and it shows you look on the right, you see the deep loess profile that we have on this landform. I've lost my cursor. And here are some of the people that are here working. Here's myself helping collect soils out of a tray. J.R. Perkins in the back left, Ralph Tucker on the upper right, and Gene Campbell's head just sticking above the soil pit. He's working away in there describing this soil. When you get on some of these lower back slope positions, sometimes it gets very shallow to the bedrock itself, and you can see the limestone bedrock here. This is pretty solid stuff in this case. Sometimes it's a little softer and you can dig through it, but in this case, it looks like it's pretty solid rock. And we can get that at various depths. In this case, it's 40 centimeters to this rock. If you move around often in the same pit, you can get depths as great as, in this case, 60 centimeters. So there's a lot of variation in the depth of bedrock, especially on these back slopes. But this gives you a little sense of what the soils look like. Often in association with that rock, we'll see that nice red or orange clayey residuum from that weathering of rock. And then we often have a veneer of hill slope sediments with coarse fragments in it, especially on those back slope positions. And this has been a great opportunity, not only for us to work with a variety of soil scientists from NRCS and forest scientists from the University of Missouri and with the U.S. Forest Service, but it's been a great teaching opportunity for a lot of our students. This is a picture of the forest soils class from the fall of 2019. There's at least 10 students shown in this picture, all of which are graduate students. And these students are actually from all over the world. We have some from Panama that were there, some from China. We have students from everywhere that come and take classes here at the University of Missouri. And this was a great opportunity for them to come and look at the work that was being done in the basket in real time. So as Mark and Kevin Godsey shown here on the right, and Garrett McKee shown here in the pit, were actually doing the work. The students were able to come, see what they're doing, interact with the professionals out in the field, ask questions, get in the pits, texture some soil, and do some wonderful things in the field. So it's been a great opportunity. And here's another picture of a pit showing all of us working together. And on the left, we have Gene Campbell, who's looks like he's comfortably sitting in a chair that he brought so he can do the recording. J.R. Perkins here next to him. Sarah Higgins, she has a poster here, and you'll hear from her in the virtual field trip tomorrow. She'll be talking about a poster that she has been working on from a research project that she did last summer out here at Basket, looking at the total phospholipid fatty acids and how they were related to land use and soils out here. Dakota Maddox is sitting behind Sarah. Dakota is a forest technician with the U.S. Forest Service Northern Research Station in my office. He's also a graduate student at the University of Missouri. And this young man here on the right, I don't recall his name, but he is an undergraduate student studying soils at the university. He had heard about this work that we were doing out here, and like Nick Jelinski said in his TED Talk, if you dig a hole, they will come. He said, hey, I want to get out there and see what you guys are doing. And he showed up and helped us one afternoon. And Garrett McKee is down here in the soil pit. That's the end of the slides that we have for you. We hope we have given you a little taste of what you might see tomorrow in the field trip. We're excited to have you with us. I'm sorry that we can't be out in the field at the same time so that we could have some big arguments about the soils and about the landforms and about the vegetation. But what Mark and I are going to do is we will be on at 9 o'clock in the morning. to briefly introduce the field trip, and we're going to replay this introduction that we're recording now. And then it's up to you guys to go through and look at each of the different videos that we've put together on the field trip. And if you go to the agenda that's posted on the website, there are links to each of those videos, and most of them are up. I think we are only waiting for one more link to be placed up, but it should be up by tomorrow morning. And that will give you that ability to go through all those different videos. They're arranged in sequence, but you're gonna, in that sequence, you're gonna learn about the basket area in our tour, but you're also gonna learn a little bit more about the Soil Health and Assessment Center that we have at the University of Missouri. We're very proud of this lab. This is one of really 2 labs in the country that are able to provide this level of soil health data and biological measures, and I think you'll be very impressed by what we have here at the university. We have 2 different tours of Sanborn Field. Sanborn Field, as you heard earlier from Donna Brandt, is a very historic and very important outdoor laboratory for us at the University of Missouri, where we've been doing work with research on crops and crop rotations since the late 1800s. So the 2 videos that tell you a little bit about the history of Sanborn Field and a little bit about what people are learning today through their work on Sanborn Field. Mark mentioned that we have the power wash videos. Mark has been doing some really interesting and some innovative work with power washing soil pits. And it's very impressive what things really pop out that you can see in a soil pit by doing a little power washing on that pit face compared to if we're just doing a normal description. So I think you'll find that video really quite interesting, and you'll pick up some tips and some things that you might want to try at home about power washing in some of the pits you're working in. We also have a very brief video of a tour of the Dooley-Miller soil erosion plots. You'll learn a little bit about that by watching the Sanborn videos, but the Dooley-Miller plots originated as a student project for someone who was working in the College of Agriculture in the late 1800s, very interested in learning about erosion and runoff. And so they established some plots on the old horticulture farm, which soon was replaced by buildings and parking lots and everything surrounding the Dooley-Miller plots because they were able to preserve them. And so you'll get a little information about those. Mark and I will be available again tomorrow at 1 p.m. to answer any questions that you have. And I think Doug Wallace will also be available to answer any questions about the ecological sites and some of the vegetation out there. So we hope you are able to tune into those videos tomorrow morning, and we hope to visit with you tomorrow afternoon. Mark, do you have anything else?
No, I'm good. I hope everybody enjoys the tour and all the other things that you can just click on and watch on demand. So have a great time. critiqued.
Hey, Mark, this is Ron. I think it was really great. You guys did a really good job on that video. I didn't get a single tick or a chigger. I think you're going in the right direction. And while I was there, I saw some of your other videos too, Mark. So I'll have to get your autograph next time I see you.
As much as we see each other, it might be a while. Thanks, Ron. Appreciate that.
Thank you, Ron. We'll give a plug to the person who actually shot the video for us. His name is Rob Mize, and he works with University of Missouri Extension. And he was a lot of fun to work with, and he was excellent in the field.
When we got down to pit 4 there, and you was seeing the hydrology begin, and Rob had his huge camera out by that time, and we was as far away from the truck as you could get. All of a sudden, he needed some help carrying that thing out. It's like, okay, here we go. But no, he was really good to work with, and he done an excellent job of capturing everything that we was trying to talk about in them soil pits and the ecological sites. Anybody else have any questions? Wow. Anybody want to know more about the biology? No?
Hey, Mark,
Yeah.
could mention that there's 2 PDF files next to the YouTube video link that has the pit information and the ecological site information.
Yes. So you can pull them down and look them over. You'll find some of the things I said in the pit and what I wrote in the description are conflicting. It's hard to remember all the details, but we had a variety of people describing, and I was trying to go in, and recap what we've seen, and I even noticed as I rewatched the video twice now that some of the things I said is not correct, and I was hoping someone would catch them. It's kind of like, Marbot Road taxonomy, and then Guy Smith, in the Guy Smith letters, he said they had asked him questions. He said, I put that in there so you'd ask a question, and no one ever did, so it's still there. I kind of like that philosophy. So yeah, I said some of that stuff so you'd ask questions, but no one ever caught it. So
Doug, you also mentioned to me in an email that you have a link to a video that you shot with the Conservation Department that explains ecological sites. So you said something about maybe you could post that in a chat for people to look at if they want to learn some more about ecological sites.
I can do that. It's about a 4-minute video, so it goes by pretty quickly. So I'll dump that in the chat right now.
is anybody thinking about going out to do some power washing? I don't blame you.
I sent you an email asking for some of the information on the camera and the software used and some of that stuff. Go ahead, and when you answer that, or if you can answer that, send me the information you've got for the power washer and stuff. How big's your water tank and that kind of thing?
I've got a 300-gallon water tank on my trailer, and I've got a new power washer that's gasoline operated, and I've got 200-foot hoses that go to my wand, and then I've got a 50-foot garden hose, so I can get a little ways from my trailer with that water source. And 300 gallons would probably be kind of minimal on a clayey soil, but it would get you there. You could get started anyway. And at Basket, there's actually a hydrant close enough. So I stretched out the garden hose and the 100-foot wand hose, and that was enough to get me over to the pit. I turned the pressure down on that to about 10 pounds, and then power wash. And on clay soil like Mexico, you'd probably want to power wash for 4 or 5 hours. And it helps if you power wash a little bit and then go take a little break and pump it out or whatever, and then come back and power wash, because that pit face kind of soaks up and then it'll wash away easier. And I'm not sure, I've never tried soap as a dispersant, but that could help too, to help disperse the clays. But I've never put soap in there to try it, so be something to experiment with. And we was thinking about a few ways maybe to try to get some real data on that root mass. One of the ways I was thinking of, and they've been using some of this with soil colors, is using a photograph. And we think that might be a way to try to get a volume on the roots. Another way was to put in nails on a grid and put them in flush with the face of that pit, and shave that off nice and flush, and then power wash to some depth, some known volume, and put the squares at a known area, and then clip them off and either weigh them or displace water, either one, to try to get some data on the actual volume or weight of the root mass. And then there's probably other ways too that I haven't thought of yet, but I'd like to get some real data on that, and look at some of these, some more of these cover crops that they're planting, to see kind of the rooting pattern of the cover crops. In the pictures at the end, the one was a Creldon soil, which has a fragipan. It's down at the southwest center. And you can see on it that after we power washed it, the only place roots were going down were in the gray seams, and all them other roots was on top of that lag line and that lithologic discontinuity on that exhumed surface. And as I washed back into the pit, they kind of lapped over the fragipan a little bit there. But as you look and take even that close-up shot of that, you can see a lot of things going on there. I like the organoargillans that you can see in that, and they go down to the top of the pan and stop. And that's probably some non-liable carbon down in there that the soil has from the days when it was in prairie, and maybe the plants have never got down there to mine that out. But them surfaces on them Creldons have been pretty well burnt out of carbon. And then the fence row was going right down the middle of two veg types, and that's why we did it on that fence row. One side was alfalfa, the other side I think was orchard grass, and that's the orchard grass roots that we're looking at on the close-up of that picture. So I did, I think, 3 soil health expos where I power washed pits. And I did that at Bradford, which is here in Columbia. I did that at Hunley Whaley, which is up near Albany. And then I did that over at the Southwest Center, which is in Mount Vernon in Southwest Missouri. So they've all been real interesting. And I learned all this from a gentleman in Illinois who was with Extension. Mike, can't remember his last name. You probably know who it is, Ron. He'd done a lot with soil health over there, and he was with the extension. And I know his first name was Mike. Anyway, we had him come over, and he showed me how to set everything up and do some power washing, and that was really good stuff. So,
Ron, you asked about the camera. All of the power washing video you did with the cell phone camera.
yeah, it was all done with a Galaxy S9, and most of it I just taped it, or I took some zip ties and tied it to a shovel handle. And then the last section of that, John actually come out and filmed me in the afternoon. And so that was all done with the cell phone camera. And I've been pretty happy with that phone. The phone I had before was it had a really good camera on it. I'm trying to remember the name of it, but I don't even think you can get that phone anymore. But the phone cameras are getting a lot better all the time, and that's good. They're really good to capture pictures with of pits. And I always like to orient the pit. You know, in the past, we always oriented pit faces towards the sun so that we could describe them well. But if you want to take pictures, orient the pit face away from the sun so that it don't cast these shadows on the pit that look weird, these angles and leaves with light coming through. It all makes it look weird. So get that thing away from the sun and make it all dark, and then the camera will do a really good job of taking pictures for you. You can even turn your flash on, which it helps on them. Get them really dark and turn your flash on, and you can take some nice pictures of pits. Another thing we was talking about and we're getting ready to do, I think, is we're gonna try to make a soil pet-on library and put all these pictures up on our website so that anybody could go in and see where they're, look at them and see what soil they are. Also, Jorge was talking about making them using GIS so that you could go to a particular spot where that soil come from, click on the point, and the profile would come up so that you could see it, which I think would be really cool. And then to hook them to an ecological site would even be more awesome, and have the taxonomy and everything there, talking about the stratigraphy, the taxonomy, the ecological site, all in one place. So I think that'd be a real great educational tool.
Hey.
Hey, Sam.
Hey, Sam.
I got a tea time. Hey, it worked good today, guys.
Good. You're able to see all the videos?
Yeah. And can you power wash my house, Mark? Well, what's good about the videos is you can use them for a lot of things now. You got it so that you just need the link, and it's a great educational tool. So, and I heard that you had the conversation about linking it to GIS. That's wonderful too, being able to go to the sites and do that. Yeah, yeah. No, I haven't started drinking yet, but tonight, John, I'll probably have a drink.
At the virtual banquet.
Yeah, virtual banquet. Yeah, yeah, yeah.
Now,
I've been trying to get my class together for Monday, so I've been working on that.
We're having a virtual banquet here.
I'm like Bob Kramer. I should have never volunteered. Yeah, yeah. So how long are you guys going to hang out there?
Well, if no one asks any questions, we're leaving.
Oh, okay. Let me think of some questions. So I saw the list of participants. That's pretty impressive. 100 and, was it almost 200?
Yeah, I think
on the list?
Over 200.
Wow. And then there were usually about 100, 110 at one time. So that's pretty good, guys. That's pretty good. So, yeah.
For those that may not notice, we did attach a attendee list to the first page of the MU website. And that's available as a PDF. We did not include contact information, just names and associations.
That's an impressive list.
If it's recorded, I bet there'll be other names added to that.
Hey, Ron.
I'm ready. Hey, I'm ready to forward that soil tour you had and with the trees. And John and Doug, I wouldn't mind seeing video tours of other ESD locations, seeing the vegetation and stuff, and linking them back to the soils. That would go a long way in helping to understand things that are going on.
John and Mark, I just want you to be aware there is a question within the chat.
Oh, that's a John question. I like it.
So it says, please reiterate the changes expected to be seen in woodland and forested soils and ecological sites over time with climate change and more frequent extreme weather events. So I'll take a stab at this, and Doug, feel free to chime in when you're ready. So one of the things that we do in the Forest Service, and it's required of national forests, with any kind of forest plan, is to also consider how that forest may change with a changing climate, and to make some sort of plans now to be managing for those trees, since trees live to be hundreds of years old. So ecological sites and soils are a great way to think about where in the landscape you might want to do some things and where you might want to do other things. So one of the things that we see at the sites like at Basket, if you have soils that are very deep, if they have a low coarse fragment content and suitable textures like silty textures, loamy textures, we know that those have very high water holding capacities. And so that gives that soil a great deal of buffering, so that when you are entering a climate where you expect to have a lot of variation, so it could be extreme rainfall, like for example, 2 days ago in North Boone County, we had 4 inches of rain come down in about an hour. So that kind of thing is expected to happen with greater frequency. So we know we have to deal with that. Similarly, we could go, in the future, we may go several weeks or months without rain. So we are expecting these greater extremes. And when you have soils with a really high water holding capacity, such that it takes 30, 40, 50, 60 days to deplete the water from field capacity to the wilting point, we know those soils are very well buffered for drought conditions. And so that we can think about those kinds of soils as places in the landscape where we might want to manage for a variety of species, some that may be a little more vulnerable to drought because they're probably not going to see as much drought stress in that kind of location. In terms of how we manage that vegetation, Doug mentioned that woodland ecological type that we're interested in managing for. And I guess what characterizes a woodland is that it has a low density in the mid-story and understory. So the way to think about it is it can be much like a forest on top of the big trees, but many of the mid-sized and small trees are absent. And that's largely because of a disturbance history that usually involves periodic fire. Top kill or remove small diameter trees, but it tends not to kill the larger trees or even reduce their growth that much either. So they tend to persist, and you have a lower density overall in that stand. And I think Doug mentioned this at one of the sites, I believe at site 1, when you start reducing the tree density, now you're starting to free up water primarily, but also nutrients that are available for the other trees that are left behind in that stand. So if we do thinning operations and some prescribed fire to reduce the density of that woodland, then we are actually making more water and nutrients available for those remaining trees. And that further keeps those trees in a better condition if a drought should come along. Now they have more water. There's less competition for those resources if you have fewer trees out there in the future. So one of the strategies that is really good in natural systems is to restore that woodland condition, that lower density condition, and some of the other characteristics associated with applying prescribed fire. Not only does that have a very rich ground floor diversity and helps conserve a lot of our native plants that we have here in this part of the world, but it also positions that stand to be much more resilient to any kind of drought situations that arise. Does that help answer your question, Rachel? Doug, you have anything else to add to that?
Just one thing on a landscape scale, if you create a mosaic of conditions where you have like refugia areas where a certain part of the landscape is really impacted by drought, but you've got maybe open landscapes where woodland species can come back in if there's some death going on. That's another management scheme that probably could help with these extreme weather events that we're going to be seeing in the future.
Yeah, and that's a great example, Doug, because that landscape that we're in demonstrates where in the landscape you have that capacity to supply a lot of water, like in the Weller type soil, or when we get in that lower foot slope position in that little benchy area, or in that bottomland. So those are great places to have the refugia. And then that Bardley soil is a much more drier soil. It has much lower water holding capacity. That's the place to really focus on those woodland species and that low density, and to select for species that are better adapted to that dry condition there. So you're matching the species to the kinds of soils, and at a broad scale in a landscape, you have a real mosaic of different environments you're managing for, just because of that soil variation in our landscapes.
Got a Zoom question about, it says, I enjoyed the tour. 3 questions. Are frangipans common at Basket? No, we don't have frangipans at Basket, even with the lithologic discontinuities. There's a little bit of cementing on the Roxana loss that we've noticed, but it's not enough to even be fragic at the subgroup. The effect on native vegetation from a fragipan, because we don't have them, we don't see that. But you might be able to address a little bit that lithologic discontinuity in there, John, and does that have an effect on the veg?
Yeah, it would have a minor effect on the veg in the sense that it affects the drainage. So that when you start changing the drainage, then you start changing the species composition a little bit. And I'll speak more generally about frangipans. In forestry, frangipans can, if you have a really strong, well-expressed frangipan and it's very continuous, it can actually decrease the productivity of the forest, meaning that it will actually produce less volume on a given acre or less biomass on a given acre. And that's largely because site and soils and soil volume has a great effect on rooting, and that in turn has a big effect on tree height. And so you tend to have shorter trees or a lower site index, we call that in forestry, on sites with the frangipan, because that pan is inhibiting rooting. Now, one thing I will say about frangipans in Missouri is that we tend to find them in some map units, not in Basket necessarily, but farther south in the Ozarks. But we often find that they're not spatially continuous. So sometimes you'll dig a pit and have a beautiful frangipan in it, but it may not extend too much farther than the pit walls itself. And often we'll find, if you dig another hole nearby, that the tree roots are getting around any kind of frangipan. So sometimes we find them in the soil, but they aren't having that effect on the forest vegetation unless they're very continuous and they're keeping roots from penetrating.
And the other thing I've noticed from working on these pans in the soils is there's a lot of tree throws, and you get some wind up because the trees don't root very deep on these frangipan soils. The tree throw is a huge disturbance on this soil. And I think that's what happened to the pans, that they're not continuous, because over the years with that many tree throws going on, that's a huge soil disturbance. And if you look at the bottom of a tree throw on a fragipan, like the 6 or 8, maybe 10 inches of that bottom of that tree root was stuck in the pan, and it flipped it up whenever it fell over. And so over the years, continuous tree throwing and degradation of the pan over the years, and pretty soon we're not seeing continuous pans across these broad ridges. And the pans formed on broad ridges, so you got to have narrow ridges won't work. You really need these broad ridges where you have the loess on top of the old truncated surface. And you'll see them lag lines over that red clay. That red clay, it seemed like it was dry, and it opened up and cracked. The loess blew and it fell down the cracks on that old truncated surface over time. And that's them gray seams that you see in there today. And then amorphous silica moved down through that profile and cemented that pan in there. So pH had to be just right. And I think you probably have to have deciduous trees.
Usually.
You have to have deciduous trees. So everything had to be just right in order for the pan to form. And then I've seen the thickest pan I've ever seen was about a 4-foot thick one down real far south in south east Missouri. And the thinnest pan I've seen is just a few inches thick. But and then like John said, I can dig a pit, and on one side of the pit I have a nice pan. On that side, over on this side of the pit, there may not be a pan. And I think the pans were continuous and they was all there at one time, and now we're just not seeing them anymore. They've degraded. But that's my opinion. So,
Hey, Mark and John, in our ecological classification system, we had 2 types of fragipan communities. One was a fragipan upland woodland, which was a little bit sloping, maybe 2 to 6% slope that had pans. And there the growth was fairly decent because we had some lateral water movement from the pan area away from the site. But we did have a fragipan upland flatwoods that was 0 to 2% slopes. And when those pans were contiguous, you had a dramatic change in composition. It was dominated by post oak, blackjack oak that were maybe 30, 40 feet tall. You had standing water in the spring, so sedges were dominant. And in the summer, they would dry out completely. So you had that wet, dry cycle within a given year, which really has an impact on the community composition. So there are parts in Missouri where pans are very well expressed, and you can see that with the vegetational response to that.
the other thing the other place I've seen pans is on foot slopes that are flat enough to keep the loess component, and on benches. So structural benches that are flat enough to keep the loess component. So you got to have that lithologic discontinuity in there. And if the loess could remain on them geomorphic surfaces, then, and everything was right, then you could get good pans. So broad ridges, flat foot slopes or toe slopes, and benches is where I've seen them. The question about Dooley Miller and getting a file not found or not being able to get there, if you Google Dooley Miller, it'll come up on YouTube. And if you Google Sanborn Field, it'll come up on YouTube. So you can find them that way if you're having trouble.
Yeah, and we noticed that at the end of one of the Sanborn Field videos, it automatically started to load the Dooley Miller.
Mm-hmm. Right. So, I want to mention too, and we're not getting a lot of questions, but what's next for basket? So we've been talking about that some, and one of the things that with 70 some full soil characterizations over this small area, we're talking about a digital soil model. And Dr. Ben Knapp's really interested in that as a research area. And so we'll probably, that'll be a project we'll be looking at. You hear that, Ralph? And then,
Thanks, Ralph.
and then we're talking about putting a scan site out there on a ridge and running two separate temperature and soil moisture sensors, one on a protected slope and one on a non-protected slope, and see if we can get some data on them protected and non-protected slopes. So hey, that's pretty cool. Look at my head.
And along those lines of developing digital soil models and so forth, education is a really important part of the basket center. And I'm thinking that the data that are used for digital soil mapping, all the vegetation data that are collected there ought to be made available for students to use to do student projects, so they can practice some digital soil mapping techniques and vegetation analysis work. But also, this might be a good training area for both digital applications and also doing field work for new employees that are in the soil survey program, NRCS. You know, one of the things, you know, we seem to press for digital things all the time, but what's really important is for soil scientists to get out in the field and see what things look like outside. And maybe the basket center and these data that we're collecting can be made a place for training.
Yeah, I'd like to encourage everybody to go out in the field and dig holes and collect, sample the soil, describe them and sample them. And, you know, we went, you know, up into 2000 and so, and the initials were all completed, and it's just like, well, we got stuck in SDJR and everybody sat down and no one's went back outside yet. And we need to go back outside. There's a lot of data that we need to collect, and we shouldn't put our shovels down and say it's done. It's not done. It's a long ways from done, and there's still a lot of work to do. And the next generations, I'm eligible. I can leave any day. So the next generation's coming, and you guys need to pick up the baton and carry it well, and go out and look and describe and sample and get this new soil biology stuff is so awesome. I mean, it's just, it seems overwhelming, but you know, if we didn't look, we would never know. And we're starting to look, and we sat down in a room and correlated this stuff with a whole bunch of people. We had microbiologists and soil scientists and geologists and geomorphologists and foresters and ecological site specialists, and we had all this data on the wall in 3 or 4 different digital forms, and we was looking at all the data at once, and people was, you know, different disciplines was chiming in on what they thought was going on, and putting all that stuff together really completes the whole picture. And you get this holistic approach to correlation that we never had before in the past. And you start to try to understand biology and what's going on, and you can make some inferences, and you can start fleshing this stuff out, and you're going to find out some things, and you're going to be like aha moments. It's like, aha, I see what's going on there now, and you know, that stuff's, to me, it makes me want to know. I want to go find out. I want to figure this out, and hopefully it would do the same for you. And hopefully through this this presentation and these tours that you've seen and some of the things that you've seen, that it would entice you to go out and look and begin to do the collection and try to understand what's going on. And I think with that, as we collect more and more data, there's going to be synergy, and that synergy is going to feed over into research and professors and universities and students and people they're going to see this data, and they're going to say, I wonder, I have a question about this, I'm going to do a project, and they're going to do their projects, and they're going to find out more and more information, and all this synergy is going to work together to help us understand how the biology is related to the chemical and physical properties of soils and them ecological sites that we are trying to frame all this in with that metadata. And all that's important stuff, and hopefully you caught the bug.
We've got a couple of questions on Zoom. You want to read that?
Let's see. With the long documentation history of Sanborn Field, have any soil health folks used those plots to evaluate methods? Yeah, and we're getting ready to do a full-blown sampling this fall on all the plots. And once,
Sanborn Field.
yeah, Sanborn Field, and once that'll be baseline data, because they're getting ready to change some of the management techniques. So as you've seen in the video, Sanborn Field has always tried to keep up with what's going on in the real world. And so no-till, minimum till, all that's going to start, you know, I think there's one plot now that's no-till, right? But they're going to start moving more plots to no-till and doing different things. So they took this full-blown analysis in '89, I think was the last time. And we've never collected biology on Sanborn Field except for Saranya's work. I did some KSAT work on Sanborn Field with a grad student, and we're going to collect full soil characterization, including biology, on all the plots, and that'll be baseline data. And in one of the presentations, I think maybe it's the one Donna did, but yes, the one Donna did where she was talking about, she was showing the results of Tucker Field and Sanborn Field. That was in that Saranya's grad work. Tucker Prairie is the reference state. And so we know the reference state, we know the baseline data on Mexico soils. And we've looked at Mexico under 25 years of restoration. We've looked at Mexico and agroeco states up on the ARS and Centralia. We've looked at Mexico at Sanborn Field and Prairie Forks as Mexico. So we've looked at Mexico under a lot of different eco states. And because we have the reference state and the baseline data, we are able to see how management has changed the biometrics over the years. And that's really interesting stuff. And one of the things that we seen was on the site that had been in CRP, four-way mix, native grass for 25 years, the fungi never returned. And one of the reasons we think it didn't return is because the surface bulk density went up when it went under the plow, and we've never got the bulk density back. And we think fungi need a lot of room. They need a porous place to live. And without that porosity, it's like you move the fungi from a mansion to this one-room shack, and they just can't live there yet. And I think it takes a long, long time to get bulk density back, your reference state bulk densities. And it's going to take a lot of roots and glomalin and all kinds of different things to make that porosity come back. And it's, even though it's a dynamic soil property, it's not one that changes very rapidly. And so we're seeing a lot of these, you know, every property is dynamic, right? They all change. It's this time reference that we got to watch, and some things just don't change very rapidly. Let's see, we got another question up here.
It's a comment.
Do we need help with digging pits by hand on those really steep sides? Yeah, we do, Gene. Yeah, we've dug what, 52?
Yeah, something like
I think we've got 52 that we could reach with the backhoe. And he's talking about the basket area. So where we can't get with the backhoe is like 35% slopes. I just won't do it. So maybe someone will, but not me. So yeah, maybe we can get some students to take some shovels and picks and go over there and dig them. And then we've got some down in the bottom that if we could get there, we could dig them with the backhoe, but we haven't found a way down in there yet. So
we can get there, we just can't get back out.
So yeah.
Looks like Gene's looking for volunteers.
Yeah,
so,
any of you students.
Go ahead and send an email to Gene when you're ready to come on down to help us. That's it. Oh, there's a new one at the bottom now. Hey, JR.
J.R. Perkins said, how do they account for the dramatic changes in corn and soybean genetics on Sanborn Field? Do the varieties of species have any effect on the soil health properties? Do you know anything about the varieties that they're using?
No, I do not know. I will say this, when it comes to the changes in the genetics, when you compare a corn yield to another, and if it's the same variety of corn, at least you'll see the rotation effect rather than the yield effect. The yield will be greater. It probably increases over time with the improvements in the breeding. But I think the reason why they're able to make the comparisons among treatments is they probably have the same varieties of corn in each of the fields and so forth. That's my guess. That's really a question for Tim Reinbach.
Tim's not on, are you?
No, Tim's not on.
Okay. Might still be planting Williams beans out there.
That's right.
Arthur wheat, Williams beans. That says a lot about our age, doesn't it? More questions? They're working on a digital soil model down on Mark Twain too, with all the work we've done down there. And hopefully it'll drape over all the Ruby Dew and Gasconade down in that area. And I think Kevin Gottsy's working on that model right now. So it'll be interesting. Is any other states doing digital soil modeling? Ron, y'all got any going on over there?
We had a couple smaller studies that we did with LiDAR and working with big data sets, but we haven't done anything on a large scale.
Curtis, did you ever come on?
Who are you looking for, Mark?
Curtis Longer.
He was on, but then it looks like maybe he got off.
Okay. More questions? So quick question. Blue lick like pedon. I'm not a loess expert, but parent material is listed as silty and clayey loess. I've never seen clayey loess. The further we get from the source, the more clay that's in the loess. So near the source, we get the coarser silts and the very fine sands falling out right near the Missouri River. And then as you move back, you'll get more silts and less of the very fine sand will fall out right near the river. And then as you get further away, all of our loess soils are fine. And so they're the clay particles are the finest particles and they blow the furthest. So the fine loess I think we got probably more soils with fine loess, or I say clayey loess, than we do with fine silty. They're just a lot broader and extensive.
like Sean's got another question.
Related to Roy's, can you talk about the loess deposit across Missouri? Yeah. You know, I think we've got a huge extent of Peorian, and that's what we see near the surface on most of our soils. And like I was saying, as far as distribution, right near the source, the Missouri River is where if you're right on the first upland near the Missouri River, we can have coarse silty soils. And then as we get further from the source, we'll go into fine silty soils, and some of the fine silty, well, some of the coarse silties don't have argillics, most of them don't. Some of the fine silties won't, but most of them do. And then as we move back further from the source, we have all fine soils, and the fine soils all have argillics. So they've been there a while. They're pretty extensive, especially across North Missouri. The flatter slopes where the loess was retained, the topography makes a huge difference because the steeper the slopes, the loess didn't stay, it eroded off. But on ridge tops and broad flats, we've got a lot of loess in the north part of the state.
Some of that's Roxana below Peoria. So
some of it's Loveland.
Peoria would be late Wisconsin, Roxana would be mid Wisconsin, and Loveland would be associated with Illinois. Is that correct?
Yeah.
So most of our delineation in Missouri is pre-Illinois.
Mark, you talked about a texture gradient. Can you also talk about a thickness gradient moving from the source out?
Yeah, so right near the source, I've seen loess deposits that are 60 feet to 100 feet thick. And as we move away, if they're good broad ridges, we can have 2 meters to 3 meters. And then in North Missouri, we'll have loess over pettus sediment from glacial outwash. And then as you get a little steeper, we'll have loess over till. And then on the real steep stuff, we might have till. And then if it gets really, really steep, we'll go into residual stuff. So the I don't know what to get, the blanket of all of them are not that thick. The further you get from the source, and erosion played a huge role in the soils that we're seeing displayed. So let's see, thought maybe clay EBT caused it as coarser. Yeah, I think that's true too. So at weathering, you can have some of these soils maybe that were deposited as fine silty soils, and then over time through weathering they become fine. And as argillics formed and the control sections filled up with clay in the argillics, and now they would be fine in the particle size class. That's true. Thanks, Roy. Yeah, we was looking at a blue lick, wasn't we, on that bench?
We're talking about it.
On that bench, that looked kind of like blue lick down there. There was no blue lick mapped out there. That don't mean that it's not there. So, yeah, good point. There's another soil called bucklick that may be out there also. Other questions? What have I been served for dinner?
I would be here. What? I would be here.
I looked at Jorge, he won't tell me, so.
Well, I tell you what we had planned originally for an in-person banquet. Someone asked what we'd be having for a post field trip dinner or something like that. We talked about having a catered barbecue, local barbecue for dinner. And then Missouri, for those that don't know, we grow a lot of white oak in this state, and we are a major barrel producer. Missouri and the state of Kentucky are probably the two single most states that have the most abundant white oak, and also the most production of barrels for making whiskey. And so we were going to do a little bourbon tasting and a little wine tasting of bourbons and wines that were aged in Missouri oak barrels so that you could get a little flavor of that. So you'll have to make a little stop at the supermarket on your way home tonight and stop and pick up some bourbon or something like that. And you can imagine what it would have been like to all be together, to enjoying the little bourbon and the little barbecue together.
Hopefully everybody enjoyed the lab tour. Donna and them done a great job of putting that together. And the next instrument we're getting ready to buy for the soil health lab is a spectrometer, so that they could build a library for Missouri, like Rich Ferguson has done at Lincoln for the whole United States. So we have 80,000 samples in storage at Sanborn Field in a building. And then that's all from soil characterization days from the soil survey days. And then I don't know how many samples we've got since, but we've got lots and lots of samples. And we'd like to build a library that's Missouri specific for our spectrometer data. And then now, I think we could do a really good job in the field of running the spectrometer and getting quick results back with our data. So that's in the works right now. So we're looking forward to that taking place.
And then Mark, you might mention that, we have one at Springfield office. Just for the one sample spectrometer.
Springfield's got the oven, the handheld spectrometer, and we've all been to the training with Rich. It was intense training, and with COVID, we wasn't able to take right off with running samples, so we may need retrain time it comes to doing it. But it's pretty intensive training if you haven't had the spectrometer training and modeling. It was good training, but it was intensive. Well, if nobody has anything else, I really miss seeing you all, and I miss standing at a pit and having everybody argue about some property. That was always fun. And but this is what we're reduced to with the pandemic. So I know if you haven't done a virtual anything, let me tell you, it takes just as much work as a face-to-face plus all the recordings. So it's not easy. It has been a challenge, and we are really grateful to Jen and the University of Missouri, Jen and Joshua and Sarah for all putting this together and coordinating everything and making it all come together. They've done a great job, and we sure appreciate that.
Thank you. We appreciate that.
And I've got to say that this has been a very well-attended meeting, even though it's virtual. We probably have double the number of people that would have been able to make it here in person. So I think that's wonderful that we've been able to reach out to so many different people, and they've been able to at least see a little bit about what things look like here in Missouri and participate in our discussions.
Even internationally.
Even internationally.
I guess that will adjourn.
Sure.
2020 National Cooperative Soil Survey Conference. Thank you everybody for your attendance and hope you want to see everybody next year in Alabama at the national meeting. Okay, we can. Oh, let me see.